All right, so this is the last section that we're going to cover of chapter 25, and it deals with peptide synthesis. So peptide synthesis is nothing more than amide bond synthesis. And the way that you make a peptide bond or an amide is by joining the N terminus of one molecule with the C terminus of another. So we're joining a carboxyl group and an amino group. And when we do that, we make a peptide bond or an amide bond. And there are many reagents available on the market to do this, but one of the most popular and most common is called dicyclohexyl carbodiamide, or DCC, and I'll just ask you to remember the acronym. So DCC, dicyclohexyl carbodiamide, which can be used to join that carboxyl group and the amino group. The mechanism of DCC coupling is pretty long, and it doesn't even cover the entire thing. There's some proton transfers that are left out. But the bottom line is that the dicyclohexyl carbodiamide ends up doing a proton transfer, which renders it more susceptible to nucleophilic attack, and you have a carboxylate, of course, which is a good nucleophile, and you end up making something that's a good leaving group. Really, actually, sorry, it's all of this that makes a good leaving group, and that's the bottom line. So it renders the carboxyl group susceptible to nucleophilic attack by the amine. You've made a good leaving group, it leaves, and you end up with an amide bond. Again, I will not ask this mechanism, will not ask, but if you want to take a look at it, feel free to do that because it will provide you with a rationale for the reaction. Now, what is the utility of DCC coupling if we want to join two amino acids together in a specific way? Because if we have two amino acids that both have free amino groups and free carboxyl groups, well, if we mix them together in a reaction flask and just treat them with DCC, we would end up with a mixture of 4 different products. We could get the R1 as our N terminus and the R2 having the C terminus. We could get the opposite, as is shown here. We could have 2 molecules of R2 joined together, and of course we could have 2 molecules of R1 joining together. That doesn't look like a very good reaction to me because nobody wants to make mixtures in organic chemistry because organic compounds are very difficult to separate. So how do we avoid making so many different products? Well, the way that we do that is by using a protecting group. Namely, we find a protecting group for our amino group, and we find a protecting group for our carboxyl group, so that we only end up with 1 carboxyl group and 1 amino group. Well, what are these amino groups? We need to find a specific protecting group that can protect our N terminus of one amino acid and the C terminus of the other. What are we going to use as protecting groups? Well, you can imagine that we're going to cover what the protecting groups are. And once you've got the protecting groups on there, then you can couple the compounds with DCC. Once you've got the compounds coupled together with DCC, you just remove the protecting groups, and now you have the amino acids in the correct order, the order that you want. Well, the first protecting group that we're going to cover is for the amino group, and it is a carbamate. So this is part of the mechanism of the formation of a carbamate, but what I want to point out is this compound here, this big compound here. This is called ditert-butyl dicarbonate. So I'll put here ditert-butyl dicarbonate. And it has several shorthand or acronyms. The one that's used in the book is BOC2O. So yeah, that's the one that we use. Or sometimes they use I don't think our textbook uses this, but sometimes they'll use T-butoxycarbonyl, like TBOC2O. But again, I think this is the one our textbook uses. So all you do is take your amino acid, treat it with BOC2O, which is this big compound here. It ends up doing a nucleophilic attack, and then after a rearrangement, you end up forming a carbamate. So I need you to know what a carbamate is. If you have a carbonyl group, it has a nitrogen on one side and an oxygen on the other, that is what a carbamate is. So you make a carbamate, as some people call it, a carbamate, and that is the protecting group of your N terminus right here. So again, tert-butoxycarbonyl, or BOC, is a popular carbamate protecting group. So again, we can use BOC2O the way that I had it written. So if you just write BOC2O, that's the same thing as writing BOC2O. Some people write TBOC2O. There's all kinds of different ways that it's written. And if you're wondering, why would you write it so many different ways? You know what? I think it's just because it's used so commonly that people just adopt kind of different little nuances to it, but they all mean the same thing. So instead of writing out this compound like this, you could just write NH, and then you can just write BOC on the end, and everybody will know that you've got the BOC protecting group. So it's nothing more than a tert-butyl ester is what you end up with. You get it, or sorry, a tert-butyl carbamate is what you end up with. This is the mechanism for the installation of the BOC protecting group. I showed you the first step a couple of slides ago. I'm not going to ask you any of this, so I'm going to put on here, will not ask the mechanism of BOC protecting. If you want to go over it on your own time, be my guest, but it's nothing I'm going to ask you on your quiz or final exam. Well, once you've got that BOC group on there, you've got to be able to get it off after the Dcc coupling, and the way that we remove it is by using trifluoroacetic acid. So this compound is trifluoroacetic acid, right? Everybody knows, hopefully knows, that this compound is acetic acid. It's the main acid found in vinegar. So if you replace the 3 hydrogens with 3 fluorines, then you end up with trifluoroacetic acid, which is the same thing as CF3COOH, as I have in the yellow highlighted area, or it's the same thing as CF3CO2H. That's just another condensed form, or we call it TFA, trifluoroacetic acid. They all mean the same thing. So you could write this, or you could just write TFA, and I'll know exactly what you mean. Trifluoroacetic acid. Trifluoroacetic acid is a very strong acid, and that serves to remove the BOC group. And what's cool about removing a BOC, if let's say you were working as an organic chemist and this is your first day in the lab, first day as a chemist, this is the reaction. If I was, monitoring you or something, this is the first reaction I would get you to do in the lab. The reason why is because this usually works with very high yields. So maybe even 100% yield. Very effective reaction and very easy to do. All you do is take and stir the BOC protected amino acid in dichloromethane or something like that. You add some TFA. You don't even have to measure it out. You just add it in with a pipette and boom. Next thing you know, all the BOC groups are gone. And the reason why this is such a great reaction is because when the deprotection occurs, so this is the mechanism of BOC deprotection, which I will not ask you. What's so great about this mechanism is that you end up forming 2 gases. You end up forming isobutylene, which is a gas, and you form carbon dioxide, which of course is a gas. So according to Le Chatelier's principle, these go up the smokestack or out the window, so to speak, so the reaction is definitely not reversible. All right, so BOC deprotection, very effective chemistry. Now, you can imagine that making peptides is big business, and so there are many ways to protect an amino group besides a BOC. There's a whole host of different protecting groups out there that organic chemists need to know. And I would say that after the BOC, the second most common amino protecting group is called an FMOC, which is made from this interesting compound here. This is called 9-fluorenyl methylchloroformate. You don't have to have the structure memorized. I would just know that this acronym is, we use FMOCCL. Okay, that's the acronym that we use for all of this. So you would react your amino group on your amino acid with FMOC chloride, 9-fluorenyl methylchloroformate, and you add the FMOC protecting group. So instead of writing all of this every time, you can just write, for example, with this one, I'll write it the exact same way. You have your nitrogen and hydrogen, and then you can just write FMOC, and everybody will know what you're talking about. It says here the installation for the FMOC is very similar to that for the Bach in terms of its mechanism. The amino group of the amino acid is a good nucleophile, but when we want to remove the FMOC, the way that we remove it is by treating it with this molecule, which is called Pipridine. And another reason why this is such a great reaction is because you end up forming carbon dioxide gas, which drives the reaction forward. Another reason why this reaction works so well, FMOC deprotection, is because in the first step, when this proton is removed, you end up making something that's aromatic. So if you remember your aromatic chemistry, it's like a cyclopentadienyl anion, and then it undergoes the rearrangement to liberate CO2 gas. So to reiterate, what I'm saying is if you want to remove the FMOC protecting group, all you have to do is treat the compound with piperidine, which is this compound. So it's like nitrogen in a 6-membered ring. And then you end up with just the amino group that is completely freed of any protecting group. So deprotection for an FMOC is piperidine. Now that we've covered 2 protecting groups for the amino groups, now let's take a look at the carboxyl groups. How do we protect those? Well, a classic way is to make an ester. And hopefully you all remember esterification reactions where you take a carboxylic acid, treat it with an alcohol and a catalytic amount of acid to make an ester. Well, the 2 most popular esters that we can make are methyl esters and benzyl esters. So in a methyl ester, your R would be a methyl group, a CH3 group, and for a benzyl group, it would be CH2 with an aromatic ring. So that would be a benzyl. So benzyl and methyl. Those are the most common. And if you're wondering, what makes these so special? It's because they're easy to hydrolyze. Very easy to bust them off just by treatment with base. So what we do is treat them with base is one option. Or if you want to remove the benzyl group, it can be removed just by treatment with hydrogenolysis. So benzyl groups just can be removed by treatment with hydrogen and palladium, platinum, or nickel, or they can be removed with HBr and acetic acid. That's something I've never done. I actually never heard of that until I read this textbook, but then I looked it up and it turns out it is a thing that I just was not aware of. So yeah, that's how we would remove these groups. But again, another effective way is just to use a base, right? Just standard saponification reaction. So we could scribble that in here. If you want to remove that protecting group, just treat it with sodium hydroxide and water, and that would give you the carboxylate. So let's scribble that in here. I'll try to make it the exact same like this. And there you go. That's how you would hydrolyze it. Again, if it was a methyl ester or a benzyl, you can use these conditions. But if you have a benzyl ester, you can remove it using hydrogenation. You can't do that with a methyl, only with a benzyl. Or with a benzyl, you can also remove it with HBr and acetic acid. The reason why people like benzyl groups so much is that you've got a chromophore in the molecule, meaning you've got something that's conjugated so it absorbs UV light. If you were to run the TLC on an amino acid that has a side chain just like an alanine, you're not going to see it on a TLC plate because it doesn't absorb UV light. But once you put an aromatic ring on there, then you can see it absorb UV light and it becomes easier to work with, just if you have another technique by which you can identify it. So that's why people like benzyl esters so much. Also, when you want to remove it, if you're only using hydrogen, well, hydrogen gas isn't strong acid, it's not a strong base. Therefore, if you have another group in your molecule that's labile, meaning it's not very, resistant to acid or base, well, then hydrogenolysis or hydrogenation would be your best choice. So you would want to choose a benzyl group. Now, how do we put all this together? Let's say we wanted to make this peptide. Okay, so let's say we just wanted to link alanine and glycine together in that specific order. You want an alanine and you want a glycine attached to it. Remember that an alanine an alanine is a is an amino acid that has what did I erase here? Anyhow, that's got just a methyl group on it, whereas a glycine the alanine has a methyl group on it, like this, and the glycine has no R group. So what am I doing here? Here, we'll just put it. Yeah, I guess I could have left it like that. There we go. Okay, so here's my two amino acids, but I want to link the carboxyl group of the alanine with the amino group of the glycine. How am I going to do that? The first thing I need to do is I need to protect the amino group of the alanine. So I'm going to do that in this step right here. I'm going to protect it as a Bach group. So now I've got the Bach group, and the reason they put it on the left-hand side is because that's where the N terminus is, right? We go NC carbonyl. That's the way we think. So now I've got my Bach on my alanine, and now I need to protect the carboxyl group of my glycine. So I'm going to make a methyl ester by treating it with acid and methanol. And now I've got my methyl ester on the right-hand side. So now on this side of my alanine, I still have my carboxyl group. And on this side of my glycine, I still have my amino group. So now I can couple them together with DCC. I'm going to erase that just because it's kind of funny looking. But anyhow, now I can bind them together with DCC, make my peptide bond. So this is the peptide bond, which is, yeah, there's the peptide bond. And then in the first step, I remove the Bach. So remove the Bach group with TFA. And then I remove the methyl ester, remove methyl ester with sodium hydroxide and water. Just a plain old hydrolysis, and that's it. Everybody with me on that? On the whole synthesis of a dipeptide, protection, coupling, deprotection. So we have to protect the N terminus of one. We have to protect the C terminus of the other. We couple them together with DCC, and then we knock off the protecting groups. All right. Well, with all that in mind, let's take a look at this. It says this method, this is a way to make a dipeptide. There's nothing wrong with this. However, it says if you want to make a large peptide, you don't want to keep doing coupling after deprotection and protection and coupling and deprotection over and over and over. It gets very cumbersome because for every amino acid that you add, you have to do 3 reactions and you have to purify it every time. So there is a way that we can synthesize a longer peptide and get very high yields. It's called the Merrifield synthesis. And the way that we do it is we tether or join the peptide that we're synthesizing to something that's insoluble. Okay, it's an insoluble polymer, and it's designed in such a way that you can actually stick or attach, I should say, or bind the peptide chain to it, and then you can knock it off in the last step. And that's what we call solid phase synthesis. So you have this polymer, which looks like little plastic beads, and the reaction is actually occurring on the surface of the little plastic beads. The plastic beads are very easy to manipulate, and if the amino acids bind to them, you can keep track of them very easily. And then when the chemistry is done, you can just remove the peptide off of the solid support or the solid phase. So here is how it works. What you do is you start by building the peptide at the C terminus. Okay, so remember, we go, let's say we have a peptide, 1, 2, 3, 4, 5, 6, whatever, something like that. Remember, the C terminus is on the right-hand side, and the N terminus is on the left-hand side. So we start at the C terminus. We're going to start here. So this is the first amino acid we would add, then 2, then 3, then 4, then 5, then 6. Then once they're all added, we remove the peptide chain from the solid support. If you're wondering, what does this solid support look like, Mr. Dion? Well, it looks something like this. It's nothing more than a polymer of styrene. So we're going to learn about polystyrene in the next chapter. But if you remove these groups, it would basically be kind of like a polystyrene molecule. So they add these methylene or CH2Cl groups to them so that it's very susceptible to an SN2 reaction. Right? This is a very good leaving group. This is an electrophilic carbon. And so the carboxylate acts as a nucleophile, and it does a nucleophilic attack on here, and you lose the chloride, and then it attaches itself to the polymer. So now that it's stuck to the polymer, what are you going to do? You're going to remove the N-Bach group, and then you're going to attach the next bead. So we've only got one on there now, and then you're going to do the next one by removing the protecting group. So there it's removed. And then you couple on the next amino acid. So you've got the second amino acid. Now you're going to break that off, and you're going to keep adding and keep adding. And once you want to remove all of these, like once you want to cleave this bond and this bond and remove it from the solid phase, you just treat it with HF, hydrofluoric acid. Now the real advantage to solid phase chemistry is the ease of purification. Like I was trying to explain, the polymer beads are just in a solution, and you just collect them by filtration. Okay, there's no need to run, difficult column chromatography and all this kind of stuff. You just grab them and keep going and going and going. And now the whole thing is automated, and you can make these giant, peptides and proteins just by adding, over and over and over on the solid phase or the solid support. So for solid phase peptide synthesis, Merrifield won the Nobel Prize. It's a long time ago, almost 40 years ago, but it's a pretty amazing way to make a peptide. So let's go over this one here. It says identify all the steps that you would use to prepare the following peptide via a Merrifield synthesis. So what we're going to do is we're going to start with the phenylalanine. Then we're going to add the valine. Then we'll add the leucine, and then we'll add the second phenylalanine. So we're going to start with the red phenylalanine, and that needs to be added to the polymer. Now, the shorthand our book uses for the polymer, why don't I start by drawing the phenylalanine? So here's my phenylalanine. Looks like this. So I like to draw it like this. So NH Bach. Okay, then I have my CH2. There's my aromatic ring, like that. And I'm going to react that with the polymer. So I want to attach it to the polymer. The shorthand for the polymer we're just going to do is this. There's the CH2Cl, and we'll just make a little black circle like that for the polymer. Okay, so now this is going to do an SN2 on this, and what are you going to end up with? Let's draw it. You have your phenylalanine, so we have to go backwards now so I don't mess it up. Let's see. We've got our Bach group on there, so NH Bach. Actually, it might be easier if I put the Bach on the end. Why don't I do that? So we'll do it like this. We'll put the Bach over here. Yeah, that'll make life a little easier. I'll just leave the chirality out of it. It's not that important. Go. And then we have our oxygen, and that's going to be attached to the polymer. Okay, all I did was rotate this bond so that I could have the Bach group on the end. All right, nothing more than that. And again, don't worry about the stereochemistry. That's neither here nor there at the moment. Okay, so now I've added the first one. This one is done. It's stuck to the polymer. Now I'm going to add the valine. What do I have to do first? I have to remove the Bach. So to remove the Bach, I'm going to treat it with TFA, trifluoroacetic acid. So that would give me the free amine. Then I'm going to react it with valine. So let's draw the structure of valine. So let's see. Our valine, I'll draw it. It doesn't matter, but I'm going to draw it like this. So do it like that. There we go. We have our isopropyl group. Put NH Bach like that. And we need our coupling agent now. So we'll put DCC. And just to make things a little bit faster and easier to understand, what I'm going to have is my polymer. Then I have my methylene. Then I'm going to have my phenylalanine, PHE. Then attached to that, I'm going to have my valine. And my valine still has a Bach group on it. So I'm going to put NH Bach on the end. Okay, so what's my next step going to be? I'm going to deprotonate that with TFA. So TFA. Then the second step is that I'm going to add the leucine. So for the leucine, I'm going to write that one in green. My leucine is going to look like this. So I've got an isopropyl or an isobutyl group on it. NH Bach. Now I'm going to end up with something that looks like this. So I've got my polymer, got my CH2, I've got my phenylalanine, G, I've got my valine attached to that. Now I've got my leucine attached here, and it has NH Bach attached to it. So next I have to rip the Bach off, so I'm going to treat that with TFA. And now I want to add the last phenylalanine. So let's do that. So our phenylalanine is going to look like this. So we're going to have our Bach group. There we go, like this. And we're going to have our aromatic ring on here. So we don't want to forget our DCC, which I forgot in the last one. I forgot to add it. So there's my DCC over there, my coupling agent. And now you've got your polymer feed, you've got your phenylalanine, you've got your valine, you've got your leucine, and now you've got your phenylalanine on the end. So phenylalanine, and it still has the NH Bach group on it. So now you've got to remove the Bach group and polymer. Kind of running out of space here, so I'm going to put step 1 to remove the Bach is TFA. Step 2 to remove the whole thing from the polymer is hydrofluoric acid. And now you've got just this. See if I can do this copy. Oops, close. There we go. And there you have it. That's how you make the tetrapeptide, by using the Merrifield synthesis. Not complicated or anything, right? Well, you have to practice it on your own, but it's just, keep going, just add and add and add. And one thing that I'll put in here, a student asked me this, I just noticed this in my notes, is that you can assume you have N-Bach protected amino acids. All right, you can just assume that you've got those, that they're there, they're at your disposal, and that you can use them as much as you want. For the next section, which deals with protein structure, this is something you'll probably cover in your biochemistry class, and you might have seen this in biology already. But there's 4 levels of protein structure that students should be aware of. And we need those to describe the 3-dimensional shape of a protein. The shape of a protein can look very complex to the point where proteins and enzymes can almost look random. However, they certainly aren't. Okay? They're definitely not random. They fold in on themselves with very specific shapes for specific reasons. So the first level of protein structure is primary structure, and that's just the sequence of amino acids. If you read the section in our book, there's a little blurb in there about sickle cell anemia, which is nothing more than exchanging, in a very big, molecule, hemoglobin. So there's one main type of hemoglobin on the planet Earth, but there are variations of it. And if you exchange one of the glutamic acid residues in hemoglobin for a valine, you end up with what's called sickle cell anemia. So, or the sickle cell trait, I should say, because in order for a person to have sickle cell anemia, they have to inherit the sickle cell trait from both parents. And that shows you that it's a very serious disease that can cause pain in the joints and the extremities, and has a lot of horrible side effects that can even lead to death. But that's just from changing one residue, so to speak. And this isn't hemoglobin. This is insulin. But the idea is, if you just change one residue in of an amino acid in a protein, it can have huge consequences. Okay, so if you look at insulin, for example, the order that all of these amino acids are linked together, that's called the primary structure. Okay, so primary structure. Now, something that you might have noticed in all of these peptides is that, let's say we had, nitrogen, carbon, carbonyl, nitrogen, carbon, carbonyl, etc. You might have noticed that we have like oxygens with negative charges. We've got positive charge here, depending on the R group. Like, let's say you have a tyrosine. Well, that's not a tyrosine, is it? A tyrosine, like this, it's got a hydroxyl on it, which can hydrogen bond. What if you have an aspartic acid, which can, form a salt bridge? But the bottom line is there's lots of opportunity for hydrogen bonding in amino acids. And so because of that, they can adopt what's called secondary structure, where they can form what are called alpha helices or beta pleated sheets. And this is nothing more than, hydrogen bonding that causes this secondary structure. If you take a look at the alpha helix, it kind of looks, it's not the double helix in DNA, that's something else, but it's held together by the same intermolecular forces. It's just hydrogen bonds between the nitrogen-hydrogen bond on one amino acid and the carbonyl on an adjacent one. So you have hydrogen bonds here, 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 so on and so forth. And that's what holds the entire thing together, right? You've got another one here, okay? But that's the idea. That's an alpha helix. And the other one is a beta pleated sheet. There's two types of beta pleated sheets. There's ones that are parallel and anti-parallel. I won't go into the whole detail of that, but what happens is you have two strands that run parallel to each other, and they form kind of this shape that looks like corrugated cardboard, and then they can kind of stack on each other even like that, which is kind of interesting. And again, it's held together by hydrogen bonds. You can see that you have, hydrogen bonds in the molecule. There's one here, you have another one here, and so on and so forth. We have all these hydrogen bonds. So primary structure is just the peptide bonds. Secondary structure, there's two types, the alpha helix and the beta pleated sheet. They're held together with hydrogen bonds. Here's some more information about the beta pleated sheet. I'll just leave that there. Then we get to tertiary structure. Tertiary structure is the result of a protein folding in on itself. So once you have a peptide that's very, very long, it will spontaneously fold in on itself. And what you'll find is that since proteins are going to be in an aqueous environment in the human body, anything that has, or any amino acid that has a polar side chain is going to be found on the exterior of the protein, whereas all the nonpolar groups like valines and isoleucines and all that stuff, their R groups are going to be pointing towards the interior, right? Because they're hydrophobic. They want to get away from an aqueous environment. Another term that you'll probably cover in biology and in biochemistry would be denaturation, which is nothing more than breaking the intermolecular forces that hold tertiary structure together. If you think about, cooking an egg, when you cook an egg, as it goes from, transparent to translucent to opaque, what you're doing is you're disrupting the intermolecular forces between the proteins in there so that they are not, what's the word I'm looking for? Anyhow, you basically just break those and they, kind of get together in a random order, and that's called denaturation. And there's different ways that you can denature a protein besides temperature. It can also be affected by pH, even force, like literally force. Like if you've ever made a meringue before, like when you beat an egg white, you're using your force of your arm to beat that. Well, just the mechanical stress from the whisk can actually break the intermolecular forces, and that's how you make a meringue, if you've ever done that. The next one is coronary structure. Once you have a protein or a peptide that has tertiary structure, so it's folded in on itself, then once it joins, so you can see you've got 1, 2, 3, 4 kind of subunits here joined together. That's what coronary structure is. And the type of forces that hold, you know, peptides together in coronary structure is the exact same forces that hold them together in tertiary structure. So that would be hydrogen bonds. It would be things called salt bridges. So a salt bridge is just an interaction between like a protonated amine and a carboxylate. So let's say you had, you know, something like this. Okay, the negative and the positive are going to be attached. What else? You have hydrophobic interactions. So hydrophobic interactions. So let's say you have an alanine residue, which has, you know, let's say you have an alanine, which has a methyl group on it, and it's adjacent or it's folded in by a valine, for example, like this, like that. You'll have London forces between the two. We call that a hydrophobic interaction. And the other one that belongs in here would be a disulfide bridge. Disulfide bridge. And we looked at disulfide bridges today. A disulfide bridge is when you have that disulfide bond that's formed between two cysteine residues. As far as the protein function section goes, we're going to skip that as well. There's all kinds of fibrous proteins. Those are things that are found in your skin and your fingernails, and so it keeps your hair, and that's why hair is so strong. That's why Rapunzel could do what she did. And the way that they provide such strength, you know, a hair fiber, for example, is by wrapping a bunch of polypeptide chains together to make something called an alpha keratin. Anyhow, an enzyme, if you've ever wondered what the heck is an enzyme is nothing more than a catalyst. Just like palladium or platinum or nickel, we think about those as catalysts, but those are inorganic catalysts. An enzyme is nothing more than a protein that's a biological catalyst. That's all it is. So it lowers the activation energy and changes the pathway of a reaction. So right now in our bodies, there are reactions that are being catalyzed by different enzymes that are taking place in microseconds. And if we didn't have those enzymes in our bodies, it could take millions of years for those enzymes, for those reactions to occur. So very important for us to live. And the last one would be a transport protein. So for example, hemoglobin is a protein, and you probably might have heard this in biology, that it has an iron bound to every heme unit, and the iron carries oxygen throughout the blood. And yeah, the heme is regarded as what's called a prosthetic group, just a molecule that's held inside the protein. But that's the entire chapter from soup to nuts.